Thank you all for attending today's webinar, and I would like to give special thanks to Multicase for inviting me to speak today and providing my experience on applying expert knowledge for in silico mutagenicity predictions. Today, I would like to begin my talk with a brief introduction on how Amgen conducts in silico analysis, what tools we utilize, 4 case studies in which we employed these expert review tools, what occurred when we repeated these analyses in the future, and a general conclusion. To start, I'd like to give a brief overview of the when and how of mutagenicity predictions at Amgen. In silico predictions at Amgen are initially triggered prior to IND-enabling studies. Here, our process development team develops their synthetic scheme and formulates predicted impurities that may occur during the manufacturing of the lead compound, as well as what reagents and intermediates will be utilized. This is typically the first time we begin to employ the in silico mutagenicity predictions. Following the manufacturing of the lead compound, actual impurities are tested and generated, and this continues throughout development and even into marketing if there are major changes in the synthetic scheme. For instance, the use of new intermediates, a new CMO is employed, or there is a change in reagents. All of these could result in new impurities or the use of new intermediates. So how do we conduct in silico analysis? Amgen has a genetic toxicology unit responsible for both the in silico analysis and in vitro, in vivo genetic toxicology work. For the in silico analysis, we utilize Case Ultra software developed by Multicase. We use 2 models as recommended by the M7 guidance, a rules-based and a statistical-based model, which is expert and BMUT. Once the analysis is completed, we generate a report for each impurity, which is submission ready if needed. Another important aspect of our testing is conducting repeat analysis. This is typically done once the final synthetic scheme is developed and a market application is being prepared. We only conduct repeats for impurities that do not have Ames data, as the in vitro data will trump any in silico data. Compounds that cannot be controlled to the TTC, which per the M7, the threshold of toxicologic concern. Any analysis that was conducted greater than or equal to 2 years. And if there were any major changes in the software. For instance, we previously utilized A7B and AT E. coli, and now we utilize BMUT. So this was a major change in the software, allowing for a more robust data set within the BMUT. In addition to this, we also conduct a weight of evidence approach. For instance, if the data is more than 2 years old, but the software is recent, and the initial analysis did not generate any alerts, we could determine that the repeat and possible change in prediction is minimal and not conduct a repeat for that molecule. Results for the in silico predictions fall into 3 categories: negative, positive, and out of domain or inconclusive. Negative compounds are quickly reported with no additional work conducted. On the other hand, positive and inconclusive out of domain results require more analysis and include expert review. Based on the review, we can either determine that the result stands as is, or we conduct, and that way we conduct a follow-up Ames, or we control the compound to the TTC to de-risk the potential impurity. Alternatively, we can de-risk these based on expert review. However, Amgen does take a very conservative approach, and typically only about 1% of the compounds are de-risked by expert review, while the remaining either have Ames conducted or control to the TTC if possible. So what are the tools that we utilize at Amgen? At Amgen, we have 2 tools. predominantly that we use. One is what's known as a similarity search, and the other is consolidator. The similarity search is a tool developed by Amgen, while consolidator is a multi-case tool which is included within the Case Ultra software. To go into more detail of these two tools, we'll start with the similarity search. The similarity search is, again, a tool developed by Amgen, and what it is, it's a database of all the genetic tox data from all the compounds tested at Amgen. That's both our API, our proprietary compounds, as well as any reagents, intermediates, and impurities that we've had developed over the years or used throughout the years. So this includes, again, in silico data, AIMS, as well as in vitro and in vivo micronucleus data. So what the program does is it performs a structural comparison of the test compound with the database to see if there are any exact matches. So as we can avoid repeating the in silico work or potentially the AIMS, or to see if the compound has any analogs or structurally similar compounds that we could potentially use to de-risk the test compound in question. Consolidator is a tool within the Case Ultra software, and this tool allows the user to search the Case Ultra database to find structurally similar analogs that could be used for de-risking the test compound. This data set is larger than the individual model data set and also provides literature references for the analogs that are generated. And this typically has available AIMS data referenced as well. So now I'd like to share with you 4 case studies that utilize both the similarity search and the consolidator tool. The first case involves impurity A. Impurity A was tested utilizing 3 models. So this is expert, A7B, and AT E. coli. Results showed a positive prediction using expert, while it was negative at AT E. coli and A7B. So why was impurity A positive for expert? So here we had 2 alerts. One was the primary aromatic amine, circled in red, and the second is a urethanes alert, circled in green. Based on these 2 alerts, we had a predicted probability of 77%, which puts it in the positive category. So what we decided to do here was do a similarity search using our Amgen tool. And when we did this search, we had 2 similar analogs pop up. One was analog 1 and analog 2. Both analog 1 and analog 2 were tested in AIMS and were negative. In addition, analog 1 shared the primary amine alert, as in impurity A, and analog 2 also shared the primary amine alert, but also had the urethanes alert. So you can see the alerts of these 2 analogs were very similar to our test compound, with no additional alerts noted. So based on the structural similarity, structural alerts of analog 2, and the fact that analog 2 was negative in AIMS, we were able to de-risk impurity A as non-mutagenic, again, based on the structural similarity to analog 2. Next, I'll go into impurity B. Again, impurity B was tested utilizing 3 models. This was the expert, A7B, and AT E. coli. Results showed an inconclusive prediction for A7B, negative for expert, as well as AT E. coli. So why was impurity B inconclusive for A7B? We had here alert 824. The alert had a calculated probability of 40%, which puts it what's called the gray zone. And in this area and this prediction probability, the software cannot determine whether it is true negative or true positive. So there may not be enough alerts to shift this prediction one way or the other, or the prediction itself is not completely clear. So it puts it into, again, what's called a gray zone, which gives us the inconclusive result. We conducted a similarity search. Nothing came up that we could utilize. So we decided to run Consolidator. So here again, we have impurity B and the positive alert. Consolidator predicted impurity B was negative. And what it did as well is it provided two structurally similar compounds with Ames data that we were able to utilize to de-risk impurity B. So here compound 4396 and 4395 had the same alerts in the same position and also had the same outlining structures. And therefore, we were able to comfortably say that impurity B was concluded to be negative or non-mutagenic based on the consolidator data and the structurally similar analogs. Next, I will review impurity C. And you can see impurity C is not very different, not too different from impurity B. So you'll see some similarities here. Impurity C was also tested in the same models, expert A7B and AT E. coli. Results here, however, had an inconclusive for A7B, again, similar to impurity B, but this also had an out of domain in the AT E. coli model. So why did we have this inconclusive and additional out of domain prediction? Since impurity B and C are structurally similar, A7B had the same alert, alert 824, which was noted in impurity B, with a calculated probability of 40.6%, again, putting it within the gray zone. In addition to this, AT E. coli not only had alert 164, which again is very similar to 824 in the A7B model, but it also had an unknown fragment. So again, based on this, no matches in similarity search, we decided to run the consolidator to try to find some structurally similar analogs. As with impurity B, we were able to dismiss alert 824 and 164 based on the negative prediction consolidator and the two structurally similar compounds that were previously noted with impurity B. Again, same alerts in the highlighted green, in the same positions with the same outlining structures. And again, Ames data was available for both of these to utilize in our report. With the unknown fragment, consolidator again gave a negative prediction and again provided examples of structurally similar analogs, which were negative, as well as the statement in blue, which we were able to include in our report. This statement reads, this fragment is present predominantly in compounds with negative experimental activity in consolidator database, therefore, it's dismissed as non-reactive. So again here, based on the consolidator analogs, the statements provided within the consolidator report and the structural similarity of the alerts between these two compounds, 469 and 2279, we were able to dismiss the unknown fragment and again conclude impurity C was likely non-mutagenic. Lastly, we have impurity D. As with the other three, impurity D was tested in expert, A7B, and AT E. coli. Results here had an inconclusive for A7B, while negative for E. coli and expert. So again, why was impurity D inconclusive in A7B? Impurity D had an aniline alert, alert 9, which gave a probability of 37.6%, again, gray zone and considered inconclusive. No matches in the similarity search, so we conducted the consolidator. In this case, consolidator predicted the compound to be positive. However, we were able to find structurally similar compounds with deactivating features, which was shared with our compound to apply this expert review and potentially de-risk impurity D. So as with impurity D, compound 746 on the top had the presence of bulky ortho substitutes, which can interfere with metabolic activation of the aromatic amines. In addition to this, and also similar to impurity D, we also had a para-substituted oxygen similar to compound 670 below, which again was predicted to be negative. So based on these two structurally similar compounds, similar deactivating features to 746 and 760, we were able to predict impurity C was likely non-mutagenic. I previously mentioned the need to repeat in silico analysis after a certain criteria were met or after some time has passed. Since impurity A, B, C, and D were tested over 2 years ago using older models, impurity B, C, and D met the criteria for a repeat, while impurity A, since it was de-risked utilizing Ames data, we decided not to repeat this impurity. Again, the Ames data will always trump the in silico data. So repeat analysis was conducted using the new BMUT model, and again, this is was substituted for the A7B and AT and coli, a larger data set with more compounds and a lot more references. And you can see when we conducted the repeat, impurity B, C, and D were negative for mutagenicity, confirming our initial expert review of these 3 impurities. In conclusion, I'd like to highlight 3 main points. First, the use of tools for expert review is critical. I myself am not a chemist by training, so these tools provide a lot of information in a short period of time, which allows me to perform these expert review and again, potentially de-risk compounds and avoid doing additional studies such as Ames, if possible. 2, as previously mentioned, the best tool for de-risking is Ames. If there is any doubt, again, Amgen takes a very conservative approach, so we tend to run Ames quite often when we have inconclusive or positive compounds, in addition to potentially de-risking. And third and most important, repeats are a must-do, particularly for lead compounds going to market, as the in silico predictions can change, and de-risking at this stage of development can lead to lengthy delays in marketing applications. With that, I would like to thank you all again for attending, and please let me know if there are any questions.
Okay, so that concludes the first presentation. Thank you, Aldo. We do have some questions coming in, but I'd like to just save those questions for the end of today's session. Let's just go ahead and move right into our second presentation for the sake of time. This way we'll have plenty of time to cover questions at the end. We're going in sort of a different direction for our next presentation. Dr. Ravi will be talking about ICHM7 beyond TTC. So Dr. Ravi is a veterinarian by education and holds a post-graduation specialization in veterinary pharmacology and toxicology. He's currently working as a preclinical expert and regulatory toxicologist at Dr. Reddy's Laboratories in Hyderabad, India for the past 3.5 years. He has 12 plus years of total experience in preclinical drug discovery and generic pharmaceutical firms. He holds a track record of excellence in toxicological risk assessment, safety, ADME, and pharmacology for NCEs, generic drugs, specially reformulated drug products, vaccines, biologics, and other products. He's involved in several IND, NDA, and ANDA 505Js and 505B2 applications. He's involved in integrated product development for global markets and toxicological risk assessment involving impurity qualification, genotox assessments, PDE, and OEL monographs. He has diverse experience of working for different markets across the world, including the US FDA, the EMA, MHRA in the UK, the TGA in Australia, Anvisa in Brazil, CFDA in China, India, etc. He has 3 international patents on his name as inventor, and he has 20 plus scientific articles published in international journals. So a wealth of knowledge on today's topic. I am very pleased to begin Dr. Ravi's presentation.
So good afternoon to everyone. Myself, Dr. Ravichandra, currently associated with Dr. Reddy's Laboratories, Idea, Hyderabad, India. So today I'll be covering in short the different concepts and ICH-M7 principles where we can think of beyond TTC levels for certain class of mutagenic impurities. So in today's session, I'll be talking in brief about ICH-M7 classification with respect to TTC and acceptable intake, and how we can categorize impurities and apply control strategies, and then on the different concepts or situations where all we can try to relax TTC for mutagenic impurities. So we'll go one by one. So this classification table is the base of ICH M7. It is depending on the two major endpoints, mutagenicity and carcinogenicity, with outcomes are either based on the experimental data or QSR. When we talk about QSR, it is involving both rule-based and statistical-based tool softwares. The compound-specific acceptable intake is applied to class 1 compounds which are known mutagens and carcinogens. So TTC is applied to both class 2 and class 3 compounds, where class 2 compounds have positive experimental data and class 3 compounds are classified based on positive QSR alert, which are not related to API. If you want to rule out this positive alert obtained in QSR, do AIMS. So based on the outcome of the AIMS test, we can further classify it to either class 2 or class 5. So the class 4 compounds also show positive QSR alerts, but these alerts are also present in either API or intermediates. So when APIs are already tested and negative for mutagenicity, the class 4 compounds are also considered as non-mutagens. So the class 5 compounds are experimentally tested non-mutagens or which have no positive alert in QSAR. So there is another class of compound which are experimentally known mutagens, but they are not carcinogens in a well-conducted carcinogenicity studies in rodents. Here they are considered as general impurities because of proven non-carcinogenicity. However, this class do not find a place in the current M7 classification table. However, in recent M7 Q&A released in June 2020, this point is covered there. So coming to the categorization and control strategy, so in continuation to my previous slide on the M7 classification table, this slide talks about categorization of different impurities and their possible control strategies. When any compound or any impurity is encountered in a drug, it is majorly classified into two angles, either mutagenic compound or non-mutagenic compound. When the compound is non-mutagenic and irrespective of the carcinogenicity results, they are controlled either by deriving the compound specific PDE or by ICH limits. So when I talk about PDE, it is either based on the ICH or the EMA guidance. So on the other side, mutagenic compounds control strategy is decided mainly based on the carcinogenicity results. If it is positive, then apply AI, which is acceptable intake, and if it is negative, again derive the PDE as I explained to the non-mutagens. So if unknown, apply a TTC. So TTC and acceptable intake. So we are talking from the previous slides. So the TTC, which is 1.5 microgram per day, is associated with a negligible cancer risk of 1 in 1 lakh population. So when they are exposed to this particular impurity over lifetime. Similarly, acceptable intake, which is derived using TD50 value obtained from a well-conducted carcinogenicity studies, are also associated with the same cancer risk of 1 in 1 lakh population. So this risk is acceptable from the regulatory point of view. So coming to the actual topic of this session. So when we come across the mutagenic compounds, do not always think of applying the stringent 1.5 microgram TTC. So there are different situations or concepts where we can really think of the levels which are beyond TTC. So few of these concepts are captured well in ICHM7, and I try to exemplify a few of them in the further slides. So the last approach, which is the read across, is already covered in the last webinar. So I'm not going to touch that topic in this session. So we'll touch on the drug is when a drug is mutagenic itself. So how do we consider going beyond TTC? So when the API itself is a mutagenic compound, then there is no need to apply 1.5 microgram TTC for mutagenic impurities observed in that particular drug substance or drug product. Not required. So in this case, a PDE, ICH or ICH limits are considered appropriate as the control strategy. So most of the cancer drugs comes under this kind of category. So moving on to the next concept, which is in the exposure duration. So this is one of the most important concepts which is routinely used in any pharmaceutical industry to relax the lifetime TTC of 1.5 microgram per day for any mutagenic impurity. So we all talked about 1.5 microgram per day as a TTC for lifetime duration exposure. So what if some drug is not given for lifetime and it is indicated only for certain fixed duration, so which are explicitly mentioned in the particular drug label. So Haber's rule talks about that, which says, so higher exposure for shorter durations are equivalent to the lower exposures for the longer durations. So this principle is to refine these thresholds based on the duration of exposure. So the major assumption here in ICHM7 is the 70 years as the lifetime duration. So all the further corrections are based on the number of treatment days against to lifetime days using this formula. So with this approach, we can relax 1.5 microgram per day threshold up to 120 microgram per day, where a drug product is indicated or treated for the less than 30 days of treatment duration. So it is clear now, based on the treatment duration, the threshold for mutagenic compound can range from 1.5 to 120 microgram per day as per this table. So this concept is also applicable to the compound specific AI, but with certain conditions. So what is that condition? So the corrected AI should not be more than 0.5% of the drug dose. So in the above formula, in place of 1.5 microgram, use this AI value and correct as per the number of treatment days in the denominator. So you will get the less than lifetime AI values accordingly. The exposure duration, this is the table, gives a general examples of drugs to categorize under different treatment durations. So one can really go through this M7 guideline for more details. Just wanted to highlight in recent Q&A of M7 released in June 2020. So the HIV drugs are moved from the earlier class of 1 to 10 years to more than 10 years and lifetime treatment grew. So it makes a significant impact on the HIV drugs. So coming to the exposure duration LTL approach case example. So one impurity is seen in a generic drug product, and no related toxicity data for this impurity, including AIMS or other genotox studies. So as usual, we ran the QSR and predicted to be equivocal for mutagenic endpoint. So expert review also not helped us to rule out this prediction. So moreover, this impurity was not seen in innovative drug product, and the levels were found higher than 1.5 microgram per day. So what would you do? We took LTL approach because this drug is indicated once a year, means only one treatment day in 1 year. So moreover, this drug is indicated in post-menopausal women. So the total number of treatment days is less than 30 days. So with this, corrected TTC which we can apply is 120 microgram per day. Coming to the next case example, a mutagenic impurity was seen in a drug X, which is indicated by oral route with a maximum daily dose of 400 mg, and maximum treatment duration is 24 months. So with this information, one can apply LTL approach as it is falling under the category of 1 to 10 years treatment duration. So the corrected TTC is 10 microgram per day for this particular impurity in that particular drug. And the respective concentration in drug is 25 ppm based on the dose of the drug. So this is one scenario for this particular impurity in this drug. So even after this correction from 1.5 microgram to 10 microgram per day, so is there any way we can further refine or relax these threshold values? So we just tried, but sometimes there can be which we will see in the scenario 2 for the same case example in the next slide. So this is a scenario 2 of the same drug and same impurity which we were talking. So in this case too, what we have done is further getting in depth to the dosing regimen and treatment duration. We observed there is a way to relax further from 25 ppm to 50 ppm. So there were actually two dosing regimens with different treatment durations. So for each respective regimen, we tried to apply the corrected TTC levels. So because of the longer treatment duration, which is 24 months, is applicable only to 150 mg per day dose, and for 400 mg per day dose, it was only 6-month treatment duration. So this was given 50 and 66 ppm levels. So conservatively, we chose 50 ppm as the final limit for that particular mutagenic impurity. So which we achieved further relaxed limit just by utilizing the dosing regimen. Scenario 1, as discussed in the previous slide, gave us 25 ppm, and the scenario 2 gave us 50 ppm. So there is a difference of two-fold relaxation. Sometimes actually this type of relaxation significantly helps to justify few mutagenic impurities where it is not possible to tighten these spec limits. So this case example proved a product-specific assessment is very critical in setting or justifying the mutagenic impurities. So next, moving on to other three concepts: the natural exposure, endogenous compounds, or the indirect mutagens, which have the threshold mechanism. So I'm covering these three concepts together, so as they are interlinked and the mechanism involved are mostly similar. So coming to the threshold mechanism, what is exactly the threshold mechanism? So before getting into case example in the next slides, So we will try to understand the threshold mechanism. So when any genotoxic chemical is present at cellular level, it may cause DNA adapt or lesions and mutations. So there can be several mechanisms involved within each cell to counter this. Example like antioxidant defense mechanisms or even attenuating these chemicals by metabolic inactivations. So the DNA repair mechanisms also contribute to suppress these toxic effects. So many of these effects, in addition to several other, can contribute to this threshold mechanism. So any compound showing threshold mechanism, the PDE is appropriate with the relevant no observed adverse effect level or no observed effect levels. So any compound which is having no threshold, either TTC or AI are appropriate. So going on to the next, the case example of natural exposure and endogenous compounds. So the case example which I'm taking here is formaldehyde. So this concept can be applied to the mutagenic impurities with case-by-case assessment. When we know that this compound is also exposed via food or by any natural means, and which it is also produced endogenously. So M7 provides this relaxation concept, like however, final limit or levels should be based on the strong scientific rationale or justification. So considering the quantitative exposure assessment of the compound via food and through the drug product as impurity, with the due consideration of pharmaceutical quality limits as well, because that is also a very important key parameter in any drug product. So formaldehyde is actually one of the such classic example which I'm taking here just to discuss further. So formaldehyde is actually a bacterial mutagen. It is positive in Ames, and also it is positive in several other genotox assays, and carcinogenic only by the inhalation routes. So when we look at the exposure through the food is up to 14 mg per day, and it is also produced endogenously in most of the cells, ranging from 878 to 1310 mg per kg. So for an adult of 50 kg, it corresponds to somewhere 43,000 to 65,000 mg per day, and it has got a very short half-life. So that has to be noted. So using the similar concept, the Burke et al. In 2018 published an article with a PDE derived as 10 mg per day. So ICH-M7 also captures the same PDE in one of the narration within the text. So the mechanism of genotoxicity is actually non-linear for formaldehyde in a carcinogenicity study, which are studied in rodents. So for such compounds, the PDE is appropriate rather than TTC or AI. So moving on to the next case example, acetaldehyde. It is also a clear mutagen and carcinogenic compound. However, carcinogenicity is only seen in the inhalation route. It is significantly produced endogenously in cellular metabolic pathways up to 360 mg per day. So in addition, some amount also exposed via food ranging 2 to 19 mg per day, depending on the dietary habits of the human. So there are actually strong evidence on the mutagenicity and carcinogenicity effects of this acetaldehyde, and they are considered as non-linear with a practical threshold. So here again, PDE is considered appropriate rather than TTC or AI for oral and IV routes. So there is published PDE for acetaldehyde in a reputed journal using the similar concept as explained to earlier to the formaldehyde. So this is one more example, the natural exposure concept. So what I'm trying to say here is, so this natural exposure concept may not be applicable for all the compounds which are exposed naturally by food. So how much is actually an important factor in any natural exposure. So glycidol is one such example. So glycidol actually is a proven bacterial mutagen and carcinogen, and it is also present in food, and the dietary intake is very low, up to 80 microgram per day. So as compared to the previous examples, formaldehyde and acetaldehyde, so the levels taken through the dietary means are very low. So hence, in this case, acceptable intake derived from a appropriate TD50 value is more appropriate. So the acceptable intake for actually the glycidol is actually a 4 microgram per day. And also it is captured in ICH M7. So, but there is no discussion of how large an increase over background is considered acceptable in ICH M7. But when we use the natural exposure approach, a detailed understanding of consumption or endogenous production is useful and critical. So in addition to an understanding of the toxicology of overexposure, and the robustness of the protective mechanism against specific toxicant is also important. So coming to the indirect mutagens, so which have the threshold mechanisms as well. So this case example is hydrogen peroxide, so which is actually a mutagenic compound. So this hydrogen peroxide also produced within the body up to 68,000 mg per day, and it is also having a very short half-life and exposed via food also. The toxic moiety of H2O2 is nascent oxygen, so it can be also called as reactive oxygen species. So as I discussed in earlier slides, so there is highly robust antioxidant defense mechanism which keep on neutralizing these ROS, reactive oxygen species, to a certain extent. So once it crosses certain threshold, cells can't withstand the load of these reactive oxygen species, so producing the genotoxic insult. So hence, these will be there will be a NOEL or NOAEL for such effects, and based on which a PDE can be derived for hydrogen peroxide. So that the same concept can be applied to any peroxide impurities or peroxide derivatives of any drugs which are seen as impurities in drugs, as they behave similar to H2O2, means hydrogen peroxide. So in my experience, we at Dr. Reddy's, so we have justified successfully one such impurity in a drug indicated for US FDA. So after we extrapolate that safe level for the toxic moiety, which is actually captured in this table. So the next concept is route specific acceptable intakes. So as we all know, as per ICH-M7, the derived acceptable intake is applicable for all routes and all subpopulations. But so this is in line with actually the other ICH guidelines. For example, ICH-Q3C, which is for the solvents, gives one toxicology limit for all the routes. So while ICH-Q3D, which is actually for metals, gives the route-specific toxicology limits for oral, parenteral, and inhalation routes. So partly maybe because of the differences in the bioavailability and the low absorption by the oral route. So however, ICH-M7 gives a provision for case-by-case risk assessment for the route-specific AI with a proper scientific rationale. So below are the two chemicals even captured in ICH-M7 with the two different AI, one for inhalation route and another for the other routes. So we may choose appropriately the acceptable intake depending on the type of drug product which we are actually handling. So the coming to the last concept, as I said, because I'm not going to take the read across AI, so I'll be just covering the metabolite of the drug concept. So the impurity of the target, like whatever we discuss, sometimes turn out to be a metabolite of the same drug. So in that case, a particular risk assessment covering that particular mutagen or metabolite So generally qualifies that impurity. So we don't have to apply a TTC further. So in quick, I covered the most of the concepts. I touched based on those. So this is just summarizing the today's session. So it is important to understand the mutagenicity and carcinogenicity outcomes for any particular compound to decide the appropriate control strategy. And as I said earlier, like expert review of any QSR output is must, and it gives us immense value to the reviewers. The product-specific assessment is equally important and critical in overall genotoxic assessment and setting the limit in a particular drug product, so which I explained through several examples. So understanding the mechanism of mutagenicity or carcinogenicity is actually a very critical step in evaluating and setting the impurities limits. So with this, I thank Multicase, especially Gianna, Dr. Suman, and Dr. Rustam for giving me this wonderful opportunity to share my experiences. So thank you so much.
Okay, so that concludes our second presentation for the day. Thank you everyone for your attention. And we'd like to just quickly move into the question and answer session. I'd like to try and focus each of the questions that have been coming in into two camps. One for the first presentation directed toward Aldo Coppi of Amgen, and the second toward Dr. Ravichandra's presentation. So we have both Aldo and Dr. Ravi on the call today. Hopefully you'll be able to come off mute if you can. And I'll go ahead and use Suman and Rustam as backups if needed. But the first question coming in is directed toward Aldo. They're wondering, what is the minimum percentage of similarity to select a structural analog? So if you can give some insight, that would be great.
Sure, no problem. Can you hear me okay?
Yeah, I can hear you great.
Yeah, great. Yeah, thank you for the question. That's a good question, actually, because a lot of the similarity searches at times, we try to use at least 80% or higher for a similarity search. Sometimes that's not always possible with the consolidator, but we do try to look at the structures in addition to the percent similarity of the structures that we're comparing it to. So again, for our similarity search, about 80% or higher. Obviously, the closer to the 100%, the better. And the same for the analogs. We try to find the highest percentage possible, but that's not always the case. There are times we do see structurally similar analogs. In regards to similarity of the alert, not necessarily the structure. So the similarity may not be even close to 80%, maybe even lower, but the alert is there. And the surrounding structures are similar.
Okay, great. Thanks, Aldo.
Sure.
So a second question that came in was about using the similarity tool in the consolidator system to show non-mutagenic compounds. Is this just simply a cover-up system to do the Ames test? I guess cover-up system. So I guess maybe just a step one before you initiate Ames, I'm guessing. If you could.
Yeah, no, absolutely. Yeah, I think that's typically our first steps. Anytime we get a positive or an inconclusive, we will always do the similarity search. We'll always run the consolidator. If we can utilize those two tools to de-risk, then we don't necessarily need to run the Ames unless the team themselves prefers to go that route. If we cannot utilize those tools, we go right to Ames, or we can control to the TTC. Some of these impurities that we test are early in the process, so the initial impurity testing that we do it in silico is just a first look to see if we have an issue. So we don't always immediately go to the Ames, but it is our preferred method for sure. But again, we do run the similarity search and the consolidator anytime we do have an inconclusive or a positive.
Okay, thanks. And another question is, have you done any random Ames testing for compounds which were ruled negative using the consolidator module to validate the system?
That's funny they asked that, because that is something we're actually considering doing, is testing some compounds that we've run and utilized consolidators. You saw the repeats that I had on my presentation. A lot of those, I believe we did run in Ames recently, and we were able to confirm. But yeah, that is something that we are looking to do, is to build our database to look at the correlation between the predictions and consolidator and the Ames. We have noticed that a lot of times when we do see inconclusive, excuse me, we do see inconclusive with the multi-case software. When we run the Ames, a lot of times it actually does trend towards negative. So that investigation we have done, but yes, we definitely would want to do some more work on how well the consolidator correlates with the Ames. Absolutely.
Okay, great. So I have a few hands raised. I'm going to move to pulling people off of mute. Jadeep Sarkar, I'm going to go ahead and pull you off mute and be prepared to verbalize your question. Okay. Hello, sir. Can you hear us? Hello?
Hello.
Hi, we can hear you great. Go ahead with your question.
Hello, Dr. Ravi. It's a question to you. Just I need to know is that when I read ICH M7 guideline and all the reference literatures, and everywhere it has been told that PDE derived values, they have been normally used for impurities, if the ICH M7 clearly tells about. Now my questions are two for you. First is, if in case we have the presentation, what you showed is that if we have another derived concept of PDE, if we should, we also have another concept additional to TTC. We can relax the TTC and we can also go for PDE. Now, for example, I have a drug that is a deemed necessary drug to be given in any disease condition, wherein on the basis of risk and benefit ratio, I find that the drug has a more of risk and less of benefit ratio, right? In case of ratio 1, I have got 0.6 or 0.7. But I don't have an alternative to that drug, and I know that that drug will have a definitely deleterious effect in the long term, but I have to use it. I don't have any drug. Normally it happens if we have a carcinogenic drug, and in that cases the drugs that are basically meant for chemotherapies, there are drugs that are metal chelates called as platinum derived drugs or some other heavy metal drug. So in that case, how will I justify PDE with a drug? I mean, from the definition of the ICH M7, it say that if we have a carryover of an API in another batch manufacturing, therein we can decide that, okay, fine, my drug is carried over to the next batch manufacturing. I can decide my limit by using a PDE. But when I already know that my drug, it is deemed necessary, it has to be used and there is no alternate. So in that case, how will I justify using PDE concept for that particular drug?
Okay. So just to summarize your question, so you're asking me like, so you have an impurity, it's Metagenic, and there is a drug which is given for shorter durations, and the risk benefit is less. So how do you want to justify beyond TTC? Is that your question?
Yes, absolutely. Absolutely. My question is, this is the question. Addition to that, the question was that if normally the PDE values that is derived in IM7, it says that PDE is normally used for the carryovers or residual as per ICH M7 if you have Q3, QC, Q3, these type of guidelines. My only context is that PDE is basically used for impurities or carryovers or residuals. So I have a drug that has less of benefit and more of risk, but I do not have an alternate to that drug. Considering the options of the chemotherapeutic heavy metal drugs, they are very carcinogenic in nature. Instead, we have read papers wherein people have died of carcinogenicity caused by those drugs as compared to the normal cancer of the people. So in that case, if my drug is falling in that category, how will I justify it with the PDE concept? And the second option is that if I'm manufacturing a batch drug, I have a B batch drug already previously manufactured. I will have a definitely residual or carryover of that B batch drug in my A batch drug. That I can justify using the PDE concept, considering this is a carryover from the previous batch I can use. But when I already have an A batch drug, when it is a very carcinogenic drug, how will I justify with the PDE concept, sir, that this amount has to be decided for 120 days lifetime if I'm not using the TTC concept and I'm using that drug for particular frame of time?
Okay. So I understood two things. So I think you are talking about the cleaning validation carryover limit, and another
Yes, sir.
which I talked to in this today's session is for the actually the drug products. Actually, the drug
Absolutely,
products.
absolutely.
Setting
Yes,
the
yes,
limit
yes,
in the
yes.
drug product. So in
Yes, sir.
setting in the drug product, the PDE is not always applied because whenever you find any metagenic impurity, it has to be first controlled as per the ICHM7. So if you are
Sure.
finding any case which I explained today, like some of the concepts where you feel like, okay, there is a shorter duration, so you can correct and apply the higher limit values. Or if you are getting any by natural means, so you can further relax based on the natural exposure range. But when you're talking about the carryover, so you will not be knowing like what exactly the next drug. So depending on the drug or so whatever the nature of the particular drug or impurity you're going to handle and which may be carry forwarded in the next drug product. So that has to be first of all controlled as per the mutagenic nature of the particular compound. So there you cannot first of all apply the PDE because in the carryover limit, because actually by default, whatever the chemical you are handling, that has to be considered. So
Right,
whatever
right,
today
sir.
I talk to you is actually with respect to the setting the limit in the drug product, because the drug specific assessment can only be done in the it cannot be done in the cleaning validation, because you will not be knowing which drug product you're going to take it up the next batch.
Right, sir. Right, sir. No issue, sir. Thank you, sir.
Yeah.
Okay, so we also have some more hands raised. So let me go ahead and pull Ravindra. I'm going to go ahead and pull you off mute.
Hello.
Hello, Ravindra. Hi, we can hear
Yeah.
you just great. Go ahead and direct your
My
question.
question is to Dr. Ravi. So, the general concept and during the regulatory deficiencies, sometimes we used to get a couple of queries like the impurity is already monograph listed, and regulators are asking us to show below TTC due to the presence of structural alert. So, did you come across such type of any case study, and how do we manage that? Because already few companies are marketing with above TTC limits, and when we are filing for new application, we are getting such type of questions. How do we manage the such type of queries?
Okay, actually that's an important question. So we also face such kind of queries from the agencies. So what your question is, already the impurity is listed in the pharmacopoeias. So the limit set in the EP again, EPR USPs is again an important factor to just note whether they are controlled above TTC or below TTC. There you will find even if the data is unknown, so based on the innovative data that might have been said. So when you have any structural alert, but it is already listed in the pharmacopoeias, so you still can claim like based on the innovative drug product, they can still consider it as a non-mutagenic. But sometimes agency may not agree with this argument. So maybe there must be some background to that. So a case-to-case assessment is actually required in this case. So it cannot be directly said whether what is your control strategy with this minimal information.
Okay,
So
thank you.
there can be
One more question.
yeah, there can be two
One more.
options, like either you can rule out that QSR alert which you got it, or just find out any experimental data is available. So if
Okay.
you're unable to find any experimental data, so either you have to justify using the innovator levels or based on the listed the pharmacopoeias, or you need to conduct a fresh aim study just to prove it is non-mutagenic.
Fine, thank you. And one more question from my side is this is with respect to dosage calculation. So let us say a drug is maximum daily dosage as per the label and monograph label information. It is given some XMG without considering the salt and all. So when we are preparing a different salt, do we need to give some molecular weight correction
Yes.
or sometimes when we go with the hydrates with different salts. So is it required to correct the molecular weight? Based on the molecular weight, do we need to go for dosage correction and accordingly TTC corrections?
Yes. So actually, you need to look at what is exactly what the drug moiety. So what is the name of the API which is captured in the DMF? So that is important because sometimes you may not capture the salt, or sometimes you capture the salt. So when you're capturing the salt content for that particular free base of the drug, it is must to apply the salt correction factor while deriving the any concentration.
Okay. I have a question from Malikarjun. I believe you have your hand raised, so I'll go ahead and take you off mute. Hello? Can you hear me? Hi, we can go ahead with your question. Hi.
Hi, this is a question for Mr. Dr. Ravichandra. The question is, our product used as a contrasting agent. The exposure is short term. It is typically 1 or 2 injections. So, and the maximum daily dose is 150 grams. So, can I approach TTC for a known mutagen impurity with unknown carcinogenicity data for such high doses? And
yes, you can apply the corrected DTC because considering the very less shorter duration, which is going to be 1 to 2 injections over a period of 1 year. So, but you also need to consider what is the lifetime exposure. If it is crossing 30 days, the respective corrected TTC can be applied. If it is less than 30 days, 120 microgram definitely can be applied.
hello.
Yeah.
Yeah, one of the impurity has the AI value of 4.6 μg. For this maximum daily doses of 150 grams, I am getting the concentration limit, which is about 0.003 ppm. So can I have to control the impurity at such low level? Because the drug is used as a contrasting agent, the exposure to the patient will be limited, maybe one time in lifetime, or sometimes if it is two.
So yeah, that's what I said actually. You can correct based on the shorter duration of treatment. So the correction stage TTC can be applied for this particular drug, considering the very shorter duration of treatment. So the complete detail of the drug and that has to be assessed thoroughly before you apply, because maybe it is indicated once in a year, but later stage it might be indicated more often at older age, which again a product specific assessment has to be done thoroughly.
Okay, so a question in a different direction is, and this could be directed at all of our panelists, what if in the case we get inconclusive in both the knowledge and statistical models? What is your, how do you proceed from there?
So this is Aldo. We've had that happen to us as well. And in that case, we again used the consolidator and the similarity search first to look for structurally similar analogs. If we cannot find that, again, we would just proceed to the AIMS if we cannot control to the TTC. So, you know, whether we get the inconclusive in one model or both, our approach to de-risking is the same.
Okay, and what is the minimum number of in silico or QSAR models that you must refer to for the genotoxic assessment of an impurity? I guess aside from the statistical and expert rule-based?
So those are the two that are required by the EB7. So that's the only two that we use. There are other models within the software that we use for multi-case that we can utilize, such as the farm models. But you know, in my experience, those two are sufficient to meet the M7 requirements. But sure, you can definitely use additional models as well.
In question and answers to M7, which were published in June, I believe, there is a specific recommendation to use additional software system in case if you're not getting conclusive results from one.
Okay. So moving back to Dr. Ravi, is the LTL approach suitable for the cohort of concern?
Actually, yeah, this is a
Like
very important
NDMA or yeah, sure. Nitrosamines.
Yeah. So as I said, like this can be applied for the acceptable intake derived into, but again, here the condition is it should not exceed beyond 0.5% of the drug dose. That is one concept. When it comes to cohort of concern, so I don't have personal experience of justifying either NDMA or NDA using this correction, but definitely one can try. But I have experience with the non-cohort of concern compounds where AAs were actually relaxed and justified.
Okay.
I've seen the question before in the list of questions, something along the line, if your drug, if your API is mutagenic, should you even worry about the impurities like NDMA and stuff? I think the answer is clearly in the M7 guidance. If your drug is mutagenic, it's not a subject of M7.
Okay. When and how to use a Bear's rule, and do regulatory agencies accept its use to increase the tolerable intake?
So Haber's rules can be used, as I said, to correct based on the treatment duration. Anyone can correct apart from whatever captured in the table. So if you want to, if you are aware of the number of treatment days, you can calculate, and further uncertainty factors can be applied once you derive the lifetime AI. So whatever the table is being given in the ICH-M7 ranging from 1.5 to 120, they carry a negligible risk of 1 in 10 lakh actually, as compared to the negligible risk associated with the TTC or actual AI without correction. So this has got one more additional factor of 10. So the risk is actually low when you apply the corrected TTC.
Okay. Usha Rani, I see you have a question, and I'll pull you off mute. Okay, it looks like there's some audio issues, so we'll move on. Sorry, my apologies. I am getting the questions ready. Okay, Anurban, Malik, I'll be pulling you off mute so you can verbalize your question. Go ahead.
Hi,
Hello.
Dr. Ravi. Yeah, can you hear me?
Okay. Yes.
Hi, Dr. Ravi. Thanks for nice presentations. I have the similar questions what earlier discussed. If the drug product is itself a known mutagen, do you think that nitrosamine impurities need to be controlled as per the guidance given to the different regulatory agencies? This is my first part. And the second part is that can we apply less than lifetime approach for the nitrosamine impurities for if the drug is carcinogenic known mutagen or if it is unknown, not mutagen? Thank you.
So I think this is a one repeat question. So when a drug itself is mutagenic, so again we need to classify what sort of the mutagen impurity it is, mutagen class it is. Either it is non-COC or COC. If your drug it again COC belongs to a COC class, so there controlling a COC impurity again will take a general path. But if your drug is simple mutagen, so there I feel some risk associated because of the cohort of concern actually they carry higher risk in any drug product. So that's the reason. So when any of your drug substance is mutagenic, that means and also it is non-COC, then you don't have to correct for the COC impurities. But if your drug is also COC and if your impurity is also COC, so then you may try. But I don't have personal experience of justifying the COC versus COC. But I have an experience of justifying like mutagen to mutagen without COC consideration. So hope that answers your question.
Okay, great. Another question is, if there are 3 class 1 impurities in the API, how do you control these impurities? Do you control under the specific AI, or do you control using the multiple impurities approach, just as class 2 or class 3 impurity? Were you able to hear the question? If anyone's able to answer, that would be great.
Okay. So when you have 3 or more mutagenic impurities in a particular drug, so you have to apply a different threshold, which is actually captured in ICH-M7 for multiple mutagenic impurities. So you have to go by that particular limit. For example, if your drug is indicated more than 30 days and within 1 year, so you have to control that limit, means all the 3 impurities within 60 microgram. But if you talk about individual impurity, it is 20 microgram. So overall, for the 3 impurity, it is 60 microgram. So you cannot cross 60 microgram when you have 3 or more particular mutagenic impurity in that particular drug substance.
That's the same experience on my end as well.
Okay, thanks, Aldo. So I have about 3 more questions, and then we can close the session. So someone's asking, if the Ames is negative, should you then move on to in vivo chromosomal aberration tests or an MNT study? If anyone can give their input on that, that would be great.
Yeah, in our experience, when we run the Ames and we have a negative, we've already assessed that there's no mutagenic potential. So it's the last thing we typically do. We do run in vivo studies as part of our gene tox package, but typically not for impurities.
I agree with Aldo. I have one more input on this. When you find any positive AIMs, so you don't have to do it in vitro chromosomal, because chromosomal aberrations are for the classogenic endpoint. So based on the output, like whether it is with and without S9, you can decide further which assay you can try to rule out, or you can try to do and rule out the further mutagenicity. It is clearly given in the ICH-M7. One can refer to that.
Okay. How many analogs do you typically need in order to dismiss an inconclusive result or a positive result in one of the 2 models used for the assessment? Any insights on that?
In my experience, we try to have a few, 2, maybe 3, if possible. If you do have an analog that's very similar to yours, I mean, that would be the only thing you would need. The important piece here, in my opinion, is having structurally similar alerts. So alerts that are in the same position, that have surrounding structures that are similar. I think that holds more weight than the number of compounds that you include for de-risking. But again, typically in my experience, we're usually able to include at least 2, if not 3, structurally similar compounds.
And in that case, do you find that the regulatory authorities accept that, or have they come back and asked to provide more analog?
To date, we haven't had them come back. So we've been okay so far. So yeah, that's usually sufficient in my experience.
If I remember correctly, that was the path suggested by some representatives of regulatory authorities, US FDA, during some of their public presentations. So I think if they are suggesting to do that, they should accept that.
Okay, and lastly, kind of a zooming out perspective. So do you think that in silico methods will replace the Ames assay completely?
In a way, they're already doing that in part of negatives.
So what is the future of this technology?
Yeah, I agree.
Well, if we are very optimistic, with or without reasons, then probably it's going to be expanded to some other endpoints. We know that the regulatory side of this is very important because that will give you justification, which will be accepted by regulators. And the regulatory side of this kind of approval is usually very slow. Second part is the availability of really good quality QSRs or technologies, which depends on the advanced technologies plus depends on the good data. And the good data, that's a bottleneck in this. So the bottom line is, if the industry will finally come up out of their, I don't want to say dungeons, but secret storages with data which can be used for everyone's advantage, that will lead to creation of very good quality QSRs, which in turn will convince regulators to start accepting those QSRs instead of the actual in vivo or in vitro tests. So if I'm talking to industrial people, it's all upon you. The moment you are ready to share your, I'm sure, very high quality data with QSR vendors, then this process will start. We'll start developing better QSRs, we'll start convincing regulators, and I'm sure that the M7 practice will be spread to some other endpoints.
Okay. Okay, well, I guess that would conclude this session. I'd like to invite everyone to visit us again for our next webinar on Tuesday, August 18th, if you're interested in taking a look inside Case Ultra for the use of ICHM7 assessments. So that will include a unique demonstration and a Q&A session in 2 weeks from today. And again, thank you to Aldo and Dr. Ravi for your time and your expertise on the subject today. We appreciate your insights, and hopefully everyone found the session useful. So wherever you are in the world, enjoy the rest of your evening or day, and we will come back together on Tuesday, August 18th for the demo and Q&A. So thanks again, everyone.
Yep.
Thank you.
Thank you very much. Thanks,
you.
Aldo, and thanks, Dr. Ravichandra.
Thank you.
Yeah, thanks.
Take care, everyone. Bye-bye.